Hello and good morning to our first lesson in the topic on transporting plants. In the previous topic we looked at transporting animals and we mostly looked at the mammalian circulatory system. Today we need to have a look, or at least start looking at, transporting plants. So the first lesson in the booklet that I have given you is entitled The Structure and Function of Plant Stems. By the end of the lesson you should be able to explain how the xylem, phloem and sclera and Charma are arranged into a vascular bundle. You should be able to explain the structure and function of the xylem, explain the structure and function of the phloem, and explain the structure and function of the sclera and Charma. Okay? So we're going to start off really basic, and something that even at A level people struggle with and they often forget is that plants are living things. Okay, so plants are living things. They are alive. Just because they're not like an animal, they're not like us, or they're not like a cat or a dog or a giraffe that's constantly moving and making noises, doesn't mean they aren't alive. They are alive, okay? And in order to survive, they need certain things. At GCSE, you learned that plants need the following things, to survive. You learnt at GCSE and even lower down in the school that plants need light for photosynthesis so that they can make glucose. They need space in which they can grow and survive and live. They need water, and they need nutrients. Now, for the majority of plants, water and nutrients come from the soil. Okay, so for majority, water and nutrients come from the soil. Now I say for the majority because things like Venus flytraps get their nutrients from catching flies, okay? Now, plants also need to grow tall in order to get light. The reason they need light is for photosynthesis, to make glucose, and that glucose then has to be transported from the leaf of the plant to all the other cells so that they can do respiration. So another important point that we're going to write down underneath this is that light is needed by leaves for photosynthesis. Okay? Full stop. The glucose made in the leaf then needs to be transported to all cells for respiration. So we've got this sort of two-way flowing of materials. You've got I'm going to just do a quick diagram so you can sort of picture it. This is on my notes, so it shouldn't be much of a surprise. So if we've got a plant, we've got its leaves here. I'm going to draw it as a nice, pretty flower. So I know you all love my artistic skills. We've got The flower, its roots are down in the soil. So these are the roots. That's a leaf. That's the stem. We've got the flower there. The water and the nutrients collected by the roots needs to be transported up the plant. So water is being transported this way. Okay? So H2O or water is going from the roots up the plant. Glucose, on the other hand, which is produced in the leaf, needs to be transported both down to the roots so they can do respiration and up to the flower so it can do respiration. So this is my glucose transport. Now, glucose is needed for respiration. Yes, plants do photosynthesis, but when it is dark at night time, plants then move over to respiration. Okay? So we can say that water / nutrients collected in the roots needs to be transported upwards through the plant. Now, water, I'm not going to write it down here, but water is needed for these hydrolysis reactions. Plants are living things, they still do condensation and hydrolysis reactions, therefore water is needed by all cells so that they can carry these reactions out. Glucose, on the other hand, glucose is transported both upwards and downwards from the leaf. So when the glucose is produced in the leaf, the glucose needs to be transported through the stem upwards and from the leaf downwards through the stem. The point I'm trying to make here, and the final thing I just want to say on this slide, is that the stem is the major transport. It's moving water from the roots up through the plant, and it's moving glucose from the leaf up and down through the plant. This is the major transport system, or the major transport area found within plants. So the final thing I want to just write here is that the stem facilitates, big word, facilitates movement of both substances. Okay, so this stem is what moves the substances around the plant, be it water upwards or be it glucose in a unidirectional manner, okay, in both ways. So we can refresh this screen. Please remember you can pause the video whenever you want if you want to catch any notes. Usually in my lessons I do try to rub the board off as little as possible, however that does become very difficult. So could we put, please, the next subheading in our notes, the structure of the stem. The structure of stems. And underline it. The structure of stems. And at this point, I'm going to go back to the PowerPoint and I'm going to nick this image from my PowerPoint, okay? I'm going to paste it in because it will be useful. You can refer back to this on the PowerPoint. It is a bit bigger, but for now we can use it like this. Okay. Now, the image here shows the structure of a stem, and the reason I use this one is because it is colour coordinated. Okay. So we can start by saying that stems have the following parts. ... The first one we're going to talk about is the xylem, and the xylem on this diagram is shown in red. However, we do use red for keyword, for important points, so I'm going to use a different colour red, okay? So we've got the xylem. And the xylem are these parts in red on this diagram. I've underlined it with a red pen on my PowerPoint, okay? Now, the xylem transports water and mineral ions around the plant. So the xylem transports water and dissolved mineral ions around the plant. So this transports water from the roots upwards. So I'm just going to add underneath there, from the roots upwards. Okay. The next one on here is the phloem. Okay, the phloem. In fact, no, we'll talk about the sclera and Charma first. So the next one is Sclera and Charma fibres. That's SCLERENCHYMA fibres. And fibres is a word I can never spell. I always get the R and the E the wrong way around. Okay. Sclera and Charma fibres are basically parts of the stem that provide strength. Okay, they're not involved in transport. So Sclera and Charma fibres provide support to the stem but not involved in transport. Okay, that is really important. People often put that the Sclera and Charma fibres are involved in moving water and sugars. No, they're not. They're not involved in transport. The final one is the phloem. The phloem, and I've underlined that in blue there. Sclera and Charma's underlined in orange and xylem's underlined in red. So just before I move on, actually, the Sclera and Charma fibres are these bits here that are in blue on the diagram. Okay, all right. Let me just get this right. Yeah, they are. So the darker blue bits are the Sclera and Charma fibres. Okay, so they're found running alongside the phloem. The final one that we need to talk about is the phloem, or at least introduce phloem. And the phloem transports solutes, so things like sugars. Okay, so transports solutes like sugars around the plant. You'll often see that the movement of substances through the phloem is said to be unidirectional. What that refers to is the fact that the sugars are coming from the leaf, they need to be transported up the plant and down the plant. So we can just add underneath here that, what colour can I use? We'll use this one. We can say that movement in the phloem is unidirectional. Okay, so the three parts of the stems include the xylem, which is shown in red on here, and that transports water and dissolved mineral ions around the plant from the root upwards. We've got the Sclera and Charma fibres, which provide support to the stem but are not involved in transport. I've coloured those in orange on our diagram because it is blue originally. And the phloem transports solutes like sugars around the plant, and the movement through those is said to be unidirectional. Okay, so we need to go through each one of these now, and we need to talk about how they're formed, what their job is, and any adaptations that they may have. So we're going to need to refresh our screen. Originally, when I was going to do this video, I did think about copying an image from my notes to then paste in. I then thought, no, I will draw it. I've now changed my mind and I am going to paste it in. So just give me 1 second. So we need to put the subheading, please. Structure. Oh, it'd help if I had a correct colour pen. Structure of xylem fibres. Structure of xylem, or xylem vessels, sorry, not fibres. The xylem are a vessel, they have a lumen, whereas things like the Sclera and Charma do not. So, as I just says, from my notes, you have got a copy of these notes. I'm going to steal this image because I can't be bothered to draw it, if I'm honest. And we're going to save it. You can see that I've drawn this anyway because it's absolutely amazing. It wouldn't be this good in any textbook. So we will save it as xylem fibres. No, xylem vessels. I keep calling it fibres. Vessels. Okay, and we can then insert that onto here so that we can refer back to it. It'd help if I use the correct image. Such a numpty today. Add image. Documents, xylem vessels. This is what we want. So we've already said, I'm just going to leave that there and we'll write round it. I've already said that the xylem transports water and dissolved mineral ions from the roots around the plant. Okay? They're formed by dead cells that are arranged into a tube. So I'm going to put in red that the xylem is formed by dead cells that are arranged into a tube. Are arranged into a tube. Okay? Now essentially what happens is that plants will stack plant cells on top of each other like building blocks, okay? So we can just start annotating this. We can use orange. So number one, we can say plant cells are placed on top of each other like building blocks. So plant cells are placed on top of each other end on end. Okay, so we've got these plant cells being placed on top of each other end on end. What then happens is that lignin is added to the cell walls and something called autolysis happens. Okay, so number 2 we can put here. Number 2, lignin is added to the walls of the cells. So lignin is added to the cell walls and autolysis happens. Okay, and we need to think about a couple of these words. So we've got lignin being added to the cell walls and we've got autolysis happening. Okay, now lignin is a waterproof chemical that stops cells from being able to lose or gain water. You see lignin in wood. Wood is dead cells made of lignin. Okay, we'll talk more about that in a moment. So lignin, and it's our first keyword, lignin is a chemical added to cell walls to make them waterproof. And I did think I was going to have space down here for autolysis, but evidently not. And autolysis, so I'm going to have to move over here, apologies. Autolysis can be defined as the self-digestion of a cell by its own enzymes. So the self-digestion of a cell by its own enzymes. By its own enzymes. Okay? Now, the tonoplast is what causes this autolysis. Now, on my diagram here, you can see that the tonoplast is this middle part. It's part of the vacuole and it contains digestive enzymes. Essentially what happens is that lignin is added to the walls of the cells to make them waterproof, and then the tonoplast breaks down. When the tonoplast breaks down, these digestive enzymes in the vacuole are released, and these digest all of the nucleus, mitochondria, chloroplasts, ribosomes. Essentially the cell sort of kills itself. The word lysis means to break down. Auto means it doesn't need an input. So this just happens by itself. Okay, so I'm going to add underneath here by saying that the tonoplast contains enzymes that cause autolysis. So what we've got so far is that the plant cells are placed on top of each other end on end, and then lignin is added to the cell walls, which I've done in green on my diagram here, The tonoplast then releases digestive enzymes which digest all of the organelles found inside of these cells. That kills the cells, okay? Once the cells are dead, the end walls are removed. That creates a single tube with a lumen in the middle through which water can be absorbed, okay? So we can put number three, and we can say that autolysis digests the contents of the cell, and end walls are removed, creating a single tube with a lumen. We did define lumen in previous lessons, therefore I'm not going to do it now. So by doing this, we've created a single tube that we call the xylem. So with the xylem, the plant places lots of cells end on end. It then adds lignin to the walls of those cells. Autolysis takes place in each cell, so the tonoplast opens in each cell, releasing digestive enzymes which digest all of the organelles found within the cell. The end walls are then removed to create a continuous tube with a lumen that we call the xylem. Now, it's important to point out here that because there's lignin in the cell walls, no water can enter or leave the xylem. It can only enter or leave through what are known as pits, P-I-T-S. You should have done about that when you spoke about the structure of cells, okay? So we can just add, and I'm going to add it at the bottom in red. I might not have enough room, but we'll try. We can say that lignin prevents water from entering / leaving the xylem, and I've run out of room, so I'm going to have to write it at the top. This is supposed to carry on from here, but it's the only room I've got. Water only leaves the xylem, through pits, P-I-T-S. Okay, so that's everything we need to know about the xylem. The xylem is used for transporting water and mineral ions around the plant from the roots. It's made up of dead cells. The way those cells are produced is you put lots of cells end on end. The lignin is added to the cell wall of each cell. Autolysis takes place, that is where the tonoplast breaks open and digestive enzymes digest the inside of the cell. The end wall is then removed from each of these cells, creating a continuous tube with a lumen that we call the xylem. The xylem does not allow water to enter or leave. Water can only enter or leave the xylem through set points known as pits. Now, I did allude to this, but before we move on, I'll mention it again. The xylem is the wood of a tree. If you look at wood, if you cut a tree in half and you see that hard woody surface, that is the xylem. The reason it's hard and woody is because it's got this lignin in it and the cells are actually dead. They're not living cells. Okay? So, that's everything you need to know about the xylem. The next one we need to talk about is the Sclera and Charma. So, could you put the subheading, please? Function of Sclera and Charma fibres. So, function of sclerenchyma fibres. Sclerenchyma fibres, and underline it. Okay. Now, lignin not only waterproofs things, but it stiffens things in order to provide support. So we can just add, and I'm going to put it in red, that lignin not only waterproofs, but stiffens to provide support. Okay. Now, sclerenchyma fibres, as we've said, provide support, but they are not used in transport. So, sclerenchyma provide support to the stem, but are not used in transport. Are not used in transport. Okay. Essentially what happens is that lignin is added to some cells, added end on end. However, their end walls are left in contact. Okay, so essentially, like we had with the previous diagram, what will happen is you'll have these cells placed end on end, like so. And I'm going to have to add cell wall to all these. Put our dividers in. We've got our cells end on end. What then happens is autolysis. What we're left with, so in fact I need to add some, so there's our nucleus of each one, there's some chloroplasts, we've got our tonoplast, which contains enzymes. Autolysis takes place, and what we're left with is a group of cells end on end that still have their end walls. Okay, lignin is also added to them, so we also need to plus. So lignin, exactly like we spoke about with the xylem. So we've got lignin being added, we've got autolysis taking place, and what we're left with are empty cells that have lignin in their cell walls, and these are what we call the sclerenchyma fibres. They do not have their end walls removed. So we've got this lignin found in the cell walls of each of these cells. The reason I'm drawing it is because I haven't done a diagram in my notes, and that is what we call the sclerenchyma fibres. Okay, that is how they remain. This is the sclerenchyma. So I'll just add that these are no, I don't want my calendar. Sclerenchyma fibres. Okay, these are the sclerenchyma fibres. So we can just add that cells are placed end on end, full stop. Autolysis takes place, and lignin added to the walls, but the end walls are left in place. So I'm going to go to red. This is the key difference, but end walls are left in place. Okay. Now, the bigger a plant gets, the more support it will need, and therefore it will need more Sclera and Charma fibres. If you're a tree, you're very, very high. If you think of a Canadian redwood, it's probably 500 foot high. If you are 500 foot tall, you need lots of Sclera and Charma fibres to ensure that your stem does not bend and you fall over. Okay. Another key point is that Sclera and Charma fibres do not contain pits because they don't transport any substances. So we can just put that Sclera and Charma fibres do not contain a lumen or pits. That's because they're not involved in transport, they are only involved in support. Okay? Right then, that's the Sclera and Charma fibres. So, lignin is used for support. Sclera and Charma fibres provide support to the stem but are not used in transport. What we get is these cells placed end on end, exactly like we have with the xylem. We get lignin being added to the cell wall, we get autolysis, but then the end walls are left in place. So we've got these building blocks that can go from the root of the plant all the way up the stem to the flower. Okay? By the end, the end plates are left in, no lumen is created because they're not involved in transport, they're only involved in support, and there are no pits, again, because they are not involved in transport. Let's get on to our last one. So we need to refresh the screen. I am going to need to nick a diagram off my notes again. Could we put the subheading, please? Structure of phloem tissue, and underline it. Okay, get rid of this because we don't want that. Get rid of you, and I want this diagram here. Look at that. Oh, honestly, sometimes my drawing skills, there's no end to how good I am at things. So if we do this, and I'll just add the annotations in. I was going to draw these originally, and then once I started recording I realised I couldn't be bothered and that it would be a travesty to not use these diagrams. So we can add an image, phloem tissue, and we can put it over here. I'm going to add those annotations back in. So this one at the top should say companion cell. Companion cell. And the one at the bottom should say cytoplasm. Okay. So the phloem. The phloem transports solutes around the plant. So we can say that the phloem transports solutes around the plant. We say that it's involved in something called translocation. Translocation. You should have done this at GCSE. Translocation. Now, translocation is defined as the movement of dissolved materials in a solution. Okay, so the movement of dissolved materials around a plant in a solution. Okay, so translocation happens in the phloem. So it happens in the phloem. So we're moving these dissolved substances. Okay, now the phloem is a weird one. Okay, the phloem consists of these cells that are alive, known as companion cells, and it consists of these sort of like semi-dead cells. I know that sounds unusual to say, they're kind of like zombie cells. They're not dead because the companion cells are doing all the living for them. But if they didn't have the companion cells, they would be dead. Okay? So, sieve tube elements are living cells which are joined end on end to form a sieve tube. So these sieve tube cells are end on end to form a sieve tube. So we can put sieve tube or sieve tube cells are living cells placed end on end to form a sieve tube. Okay, a sieve tube. So we've got these cells placed end on end to form a sieve tube. In between the sieve tube cells are these sieve plates. These are essentially pieces of tissue with tiny holes in them that only allow certain substances through. Okay, they allow solutes to pass through them. So on my diagram on here, these grey parts are solutes. Okay, they're solutes. So we can put, and we need to mimic this colour the best we can. It's like that colour in it. We can say that, oh God, no, it's not that one. That's close, look at that. Sieve plates separate sieve tube cells and have tiny holes in them and have holes in them that allow solutes through. Okay, so we've got these sieve tube cells that are placed end on end. They're living cells. They form what's known as the sieve tube. Between each sieve tube cell, we've got these sieve plates which have tiny holes in them that allow solutes to pass through. Okay, sieve tube cells share the organelles of companion cells. So these sieve tube cells, they're living because the companion cells have all of their organelles. So a sieve tube in itself does not have a nucleus, it does not have a mitochondria, it does not have a ribosome. However, it shares the nucleus, mitochondria and ribosomes of these companion cells. So I'm going to put in red that sieve tube cells do not have organelles of their own comma, but share the organelles of companion cells. Okay, so they don't have organelles of their own, but they share the organelles with companion cells. If sieve cells didn't, or sieve tube cells didn't have these companion cells, they would die. Okay? Now, one final thing that's very important that we need to write down is that the phloem is only used for transport. It is not used for support. Okay? So the phloem is only used for transport, comma, it is not used for support. So the phloem only transports dissolved substances. Remember, in the phloem, it will be unidirectional. So we can also put here, and I'm going to use a colour that we haven't really used, transport in the phloem is unidirectional. If you think back to the start of this lesson, the flower I drew, we said that the sugars are transported both up and down from the leaf, whereas the water and mineral ions are only transported up from the roots. So the structure of the phloem, it consists of these sieve tube cells that are alive, unlike in the xylem where they're dead. Separating the sieve tube cells are these sieve plates, which have tiny holes in them to allow solutes through. The sieve tube cells do not have organelles of their own. Instead, they share organelles with these companion cells. It's important that it does this because the movement of sugars inside a plant is an active process. Sometimes you want to move the sugars up, sometimes they want to move the sugars down. Whereas the movement of water is not really active, it's by transpiration. Transpiration from the leaf pulls water through the xylem so that the cells don't need to be alive for that, you just need a straw. Whereas with the phloem, we do need these cells to be alive because it's an active process. The phloem's only used for transport, it's not used for support, therefore there's very little if any lignin used, and the transport in the phloem is unidirectional. So, from this lesson, you should be able to now tell me how the xylem, phloem and sclera and Charma fibres are arranged into a vascular bundle. This diagram here, that is your vascular bundle. You should be able to tell me the structure and function of the xylem, phloem and sclera and Charma. Moving forward in this topic, we're going to look at how these substances are actually transported. If you want to read ahead, that is fine. But from today's lesson, I just want you to understand the structure of each of them. I don't want you to worry about how water's actually moved and how sugars are moved. We're going to do entire lessons on that. So if you have any questions, please drop a comment in the Google Classrooms or send me an email and I will get back to you as soon as I can. Thank you.